Good day everyone and welcome to the G7, the Science, Technology and Society webinar video.
We are the BS Forestry second year students and our topic is all about biotechnology and its application in forestry.
Karen M. Ibanez, together with me.
Guys, I'm Kevin Ibarita.
Hi, I am Joanne Sumosog. I am Rose Jean Palok.
Everyone, my name is Bernie K. Alanyabu.
Hi, I'm Angelica Amper.
Hi guys, I'm George Raby Caravana.
I'm Jean Marimat and we are the group 2.
What is biotechnology? Biotechnology is the use of biology to solve problems and create useful products. It involves using living organisms, cells, or biological process to develop products that improve human life. One of the most important areas of biotechnology is genetic engineering, which allows scientists to modify genes to produce useful substances such as medicines and therapeutic proteins. So now, biotechnology uses living things like bacteria, yeast, plants or animal cells as tools. Instead of relying only on machines or chemicals, scientists use biological systems because they can naturally produce complex substances. Through genetic engineering, specific genes can be added or modified or organisms that can produce products like insulin or other medical treatments. Now, in history of biotechnology, humans have been using biological process for about 10,000 years. Beginning of the early agricultural communities, around 6,000 years ago, people started using microorganisms to make bread, alcoholic beverages, and cheese. These practices improved food production and preservation, although people that time did not understand science behind them. This early form, biotechnology, was based on observation of experience. People learned that certain natural processes could control for their benefits even without scientific knowledge of microbes. Modern biotechnology began in 1960s and 1970s, which advanced molecular and cellular biology. Scientists learned that how manipulate DNA, leading to a development of genetic engineering. During the mid to late 1970s, the biotechnology industry emerged with companies such as Genetic commercializing combination DNA technologies. This period marked the start of biotechnology as scientific field. Instead of using natural process, scientists could now directly control modified genetic materials. Recombinant DNA technology became the dominant technique of biotechnology. It involves inserting genes that produce a useful protein into cells such as bacteria or yeast. These cells then produce a protein of large amount of. In 1982, genetically engineered insulin became the first biotechnology product approved for medical use, followed by many other proteins-based medicines. This success showed that biotechnology could produce safe and effective products. It also led to rapid growth and research industry and application of biotechnology worldwide.
Meristematic tissues are found in the growing regions of the plant, such as the tips of shoots and roots. These tissues are made up of actively dividing cells, which means they have a high ability to grow and develop into a complete plant. Young plant tissues are preferred because, first, they grow faster compared to mature tissues. Second, they have a high regeneration potential. Third, they are usually free from diseases and viruses. Fourth, they maintain the genetic characteristics of the parent tree. So first, let me explain what is genetic modification. So, genetic modification is a technique that involves altering the DNA of genes to enhance desirable traits. These traits may include resistance to pests and diseases, faster growth rate, and tolerance to drought or poor soil conditions. By modifying specific genes, scientists can develop trees that are more resilient to environmental stress. For example, genetically improved trees can survive longer during dry seasons or resist insect attacks, which is very useful in plantation forestry and reforestation programs. And the second important technique is tissue culture. So, tissue culture is the process of growing plant cells or tissues in a sterile laboratory environment using artificial nutrient media. This method allows the rapid multiplication of trees that have superior traits. In forestry, tissue culture is especially important because many tree species take a long time to grow and reproduce naturally. Through tissue culture, thousands of identical and high-quality planting materials can be produced from a single parent tree within a short period of time. Now, this brings us to the question, which part of the plant is commonly used in tissue culture to grow a new tree in the laboratory? So the answer is meristem or young plant tissues. So the part of the plant most commonly used in tissue culture is the meristem or other young plant tissue such as shoot tips, buds, and nodal segments mature to observe their traits. According to Collard and Mackell in 2008, molecular markers increase the accuracy and speed of tree improvement programs. So, in conclusion, biotechnology plays a vital role in modern forestry through genetic modification, tissue culture, and molecular markers. Among these techniques, tissue culture relies heavily on the use of meristem or young plant tissues, as these parts have the highest capacity to regenerate into a new tree, and this method ensures the production of healthy, uniform, and high-quality planting materials that support sustainable forest management and conservation. Last but not the least, they maintain the genetic characteristics of the parent tree. When a small piece of meristem or young tissue called an explant is taken from a healthy tree, it is placed in a sterile culture medium, and this medium contains nutrients, sugars, vitamins, and plant hormones that support growth. Over time, the explant develops into shoots and roots, eventually forming a complete plantlet. Once the plantlet is strong enough, it is transferred to soil and gradually exposed to natural conditions through a process called acclimatization.
Biotechnology plays an important role in disease diagnosis and management in forestry. Using DNA-based tools, scientists can detect tree disease early, even before visible symptoms appear. This allows faster action to prevent the spread of infection in forests. Biotechnology also supports the use of biocontrol agents and beneficial microbes to protect trees from pests and diseases while improving soil health. These methods reduce the need for chemical pesticides. Overall, disease diagnosis and management show how biotechnology helps maintain healthy, productive, and sustainable forests, reinforcing the importance of biotechnology in a modern forestry. One of the major applications of biotechnology in forestry is tree improvement and breeding. Through biotechnology, scientists can identify trees with desirable traits such as faster growth, pest resistance, and tolerance to harsh environmental conditions. Using molecular markers, these traits can be detected at an early stage without waiting many years for the trees to mature. This makes tree breeding faster, more accurate, and more efficient compared to the traditional methods. Overall, tree improvement breeding show how biotechnology supports sustainable forestry, forest conservation, and increased productivity, which are the key goals discussed throughout the webinar.
So, in vitro propagation, also known as micropropagation, is an important application of biotechnology in forestry. It involves growing plant cells, tissues, or small plant parts in a sterile laboratory environment under controlled conditions. This technique allows foresters and researchers to produce large numbers of trees efficiently, regardless of season or environmental limitations. Next is mass production of elite clones. So one major advantage of micropropagation is the mass production of elite clones. From a single superior parent tree, one that shows desirable traits such as fast growth, straight stems, or disease resistance, thousands of genetically identical trees can be produced in a short period of time. These clones ensure uniform quality, predictable performance in the field, and disease-free planting materials, making micropropagation highly valuable for large-scale reforestation and commercial plantation forestry. Lastly, conservation of endangered species micropropagation also plays a crucial role in the conservation of rare and endangered tree species. Species that are difficult to propagate through seeds or are at risk of extinction can be grown and maintained in laboratory conditions using tissue culture techniques. Once established, these plants can be gradually acclimatized and reintroduced into their natural habitats. This approach helps preserve genetic diversity, supports forest restoration efforts, and contributes to long-term biodiversity conservation.
Genetic engineering in forestries. Genetic engineering is a powerful tool in forestry, allowing scientists to make targeted changes to the tree DNA. Transgenic trees for enhanced traits and CRISPR and gene editing potential. Transgenic trees for enhanced traits. Some traits are genetically modified to resist insect or tolerate poor soil conditions. These trees can help reduce pesticides use and improve forest productivity. CRISPR and gene editing potential. Some trees are genetically modified to resist insects or tolerate poor soil conditions. These trees can help reduce pesticides use and improve forest productivity. Offers faster, more prices alternative to traditional breeding, promising to a secure future forest resources, enhance ecosystem services, and boost the economy while tracking pressing environmental challenges. Which biotech method is used to clone?
So for our last question, name one benefit of using molecular markers in tree breeding. Well, the answer is faster and more accurate selection of desirable traits. That's all.
I will present case studies that show the local relevance of biotechnology in the Philippine context. This example demonstrates how biotechnology contributes environmental protection and sustainable development in the country. First is mangrove restoration. In the Philippines, mangroves are essential in protecting coastal areas from erosion and strong storms. Through biotechnology, scientists are able to propagate healthy and resilient mangrove seedlings using tissue culture and genetic screening. These methods increase the survival rate of mangroves and strengthen coastal ecosystem, which are vital to both the environmental and coastal communities. And the second is bamboo propagation. Bamboo is an important resource in the Philippines for construction, livelihood, and environmental conservation. Using in vitro propagation techniques, bamboo planting materials can be mass-produced efficiently. This approach supports sustainable resources management by ensuring a continuous supply of bamboo while reducing pressure on natural forests. Lastly, dipterocarp conservation. Dipterocarps dominate Philippine rainforests, but many species are threatened. Biotechnology supports their conservation through seed banking and tissue culture technologies, which help preserve genetic diversity and assist reforestation efforts. These methods are widely supported by research institutions in the country. In conclusion, these case studies highlight the significant role of biotechnology in addressing environmental challenges in the Philippines by supporting mangrove restoration, bamboo propagation, and dipterocarp conservation. Biotechnology contributes to environmental sustainability and national development.
Global innovations in forestry biotechnology show how the scientific tools discussed earlier, such as genetic modification, tissue culture, molecular markers, and biocontrols, are applied in real-world situations to solve major environmental and economic problems. This innovation helps improve forest productivity, protect ecosystems, and support sustainable development across different countries. One important global example is the genetic improvement of poplar trees of biofuel production. Poplar trees are fast-growing and widely used in forestry research. Through genetic engineering, scientists have modified poplars to produce higher biomass, which means they grow faster and yield more plant material. This makes them ideal for renewable bioenergy, reducing dependence on fossil fuels and helping address climate change. This innovation directly connects to earlier discussion on genetic modification and gene editing, showing how altering specific genes can improve tree performance for environmental benefits. Another major global innovation is the use of biotechnology to combat invasive forest pests. Invasive insects such as bark beetles and emerald ash borers have caused widespread damage to forests in North America, Europe, and Asia. Biotechnology provides solutions by using DNA-based tools to detect pests early and by developing pest-resistant tree varieties. In addition, biological control methods such as using natural predators, fungi, or bacteria help manage pests without relying heavily on chemical pesticides. This connects to the earlier topic of disease diagnosis and management, emphasizing how biotechnology promotes environmentally friendly forest protection. This global innovation demonstrates how biotechnology supports sustainable forestry. By increasing tree productivity, forests can meet demand for timber, energy, and other forest products without excessive deforestation. At the same time, protecting trees from pests and disease helps conserve biodiversity and maintain ecosystems balance. Furthermore, global forest biotechnology initiatives often guide local applications, including those in the Philippines. Technology used to improve poplars or manage pests abroad can be adapted for local species such as mangroves, bamboo, and dipterocarps. This highlights the importance of global research, collaboration, and knowledge sharing. Global innovation in forestry biotechnology bring together all the concepts discussed in this topic, from laboratory techniques to environmental application. These show that biotechnology is a powerful tool for addressing global challenges such as climate change, invasive species, and resource sustainability, while also emphasizing the need for responsible use and proper regulation. This naturally leads to the next discussion on ethical, environmental, and policy considerations.
Biodiversity, these are words about the loss or damage to the variety of living organisms, plants and animals, and microorganisms in our ecosystem. These forests Biodiversity concern means that new technology or practice might reduce native species, disturb habitats, or cause from species to disappear. Genes flow. Genes flow is the movement of genes from the population of others. In biotechnology, this refers to modified genes spreading from genetically modified plants to wild or natural change their threats or affects ecosystem. Public perception. Public perception is how people think and feel about an issue. In biotechnology, it refers to public acceptance or opposition, often influenced by concerns about safety, ethics, environmental impact, and lack of information. Ethical, environmental, and policy consideration. Risk. Loss of biodiversity. Native species may be reduced. Unintended spread of modified genes or wild plants. A new long-term effect on ecosystem. Controversies, public concern, and low acceptance of genetically modified organisms (GMOs). Ethical issue about altering natural organisms. Debate over who benefits companies versus communities.
Global agreements regulate the safe development and use of genetically modified organisms to protect human health and the environment. Our Secretariat of the CBD 2000. In the Philippines, biosafety regulations are implemented through the Department of Science and Technology National Committee on Biosafety of the Philippines, or DOST and CBP, following the international standards like the Cartagena Protocols on Biosafety.
Thank you for listening, and we hope you learned something. That's a wrap. Bye!
Thank you for your time.
Thank you everyone for being here.
Thank you for your cooperation. Bye!
Thank you for listening, guys, and bye.
Thank you everyone.
And goodbye.
Thank you for watching, everyone. Thank you for joining the webinar.
Bye-bye.